cut this part out because that's not what I meant to say, and I can't pronounce any of these words. Welcome to Life in Biology. I am your extremely unqualified guest host today, and I will be telling you about type 4 hypersensitivity. So, first of all, what is type 4 hypersensitivity? So, type 4 hypersensitivity is a type of cell-mediated immune response that has a delayed reaction, meaning that it can take up to 2 to 3 days to develop. So it's different than the other, obviously there's a 1, 2, and 3 type sensitivities, and it's different from those types because it is purely cell-mediated. So this type of reaction arises in diseases that you can see, such as multiple sclerosis, Crohn's disease, and in allergic contact dermatitis, which we're going to get more into later. Type 4 hypersensitivity was first observed in 1980 when Robert Koch observed that individuals infected with a bacteria called Mycobacterium tuberculosis developed a localized inflammatory response when injected with a filtered mycobacterial culture. So type 4 reactions are usually characterized by their initiation by T cells, a delay required for the reaction to develop, and the recruitment of macrophages instead of neutrophils or eosinophils. So the most common type of type 4 hypersensitivity that most of us know about is exposure to the toxicodendron species of plants. So that's poison ivy, poison oak, or poison sumac. And its toxicodendron sensitivity affects probably between 50 and 70% of the adult population in the US. And some documented cases have been so severe that they require hospitalization. It affects between 10 and 50 million Americans per year, and it's a significant occupational hazard. So let's talk about what happens during an allergic contact dermatitis reaction. So an ACD reaction can be also caused by exposure to like nickel in jewelry, precious metals, certain fragrances, and also chemicals that can be found in cosmetics and hair dyes. So there's 2 stages to a type 4 reaction. The first is the induction phase, where the immune system is primed and sensitized, and the second is the elicitation or effector phase. Yeah, I have to remember that. So once an allergen is exposed to the body, the T cells then utilize TNF alpha along with interleukins 1, 13, and 18 to promote or inhibit mobilization of the Langerhans cells. Yeah. In the lymph glands, dendritic cells present the allergen's epitope to T cells, and the T cells mount an appropriate immune response and build memory in order to respond to the allergens more quickly next time they're exposed. So in the case of toxicodendron plant species, they contain a compound called urushiol. I don't know if I'm saying that right, urushiol. And that's a compound that can bind to human and some higher primate skin proteins. So someone who has been exposed to urushiol develops a weeping, burning rash within 24 to 48 hours of exposure. This compound is easily degraded in water, but because it's a delayed immune response, a lot of people don't know that they've been exposed. So let's talk about sensitization and effector phases a little bit more, because that's something that is kind of important. So let's talk about the 2 phases of allergic contact dermatitis immune response. So in the sensitization phase, an antigen-presenting cell, such as a macrophage or Langerhans cell, binds to a cell such as a CD4 Th cell, or it could be a Th17, Th2, or a CD8 cell. That's not very common. It's usually a CD4 cell. And based on that interaction, the cell will then release Th1 cells to build a memory response. So that brings us to the second stage, I hope you can see this, yeah, the elicitation or effector stage, in which the now sensitized is what it's called, Th1 cell then binds to a macrophage and kind of like educates it. You know, it's what we've learned before, but and then the macrophage then develops TNF receptors, the appropriate TNF receptors, and displays, if it's a CD4 cell, they'll display MHC2. And that macrophage is now called an activated macrophage. And then it'll go off and find what it's supposed to find and take care of the rash problem. Anyway. So there was a team from China that published a paper back in December of 2015, so not too long ago, that looked at the effects of a medicinal Chinese herb with the scientific name Radix sapochnikovia, which the Chinese common name for it is, I'm going to totally butcher this, but fangfeng? I don't know. Anyway, it's a plant, and they looked at it because it's commonly used in Chinese medicinal formulas to treat allergenic and inflammatory diseases. But they didn't really know the mechanisms of why this plant worked on, like rashes and stuff, or, how it worked. So the purpose of their study was to look at, we're just going to call the plant fangfeng. We're going to call it RS, Radix sapochnikovia, because I don't want to say that a billion times. So they were looking at the effects of RS on allergic contact dermatitis in lab mice, of course, and to look at the underlying mechanisms of how this plant worked. Yeah, so what they wanted to do is look at the activation of T cells and T cell differentiation, which is mediated by dendritic cells. As we kind of talked about earlier, I tried to describe it, but sorry if it's not the best explanation. So it just says, in this study, we explored the relationships between the effects of RS extract, the activation of T cells, and T cell differentiation as mediated by dendritic cells. They then addressed this issue by investigating the effects of RS extract on DNCB-induced allergic contact dermatitis by observing the pathological changes and the changes in inflammatory cytokine expression, TH subsets, and expression of TH-related transcription factors that occur after treatment. They also looked at the surface marker CD11C and co-stimulatory molecules of dendritic cells during the treatment with RS extract. So what they did is they took BALB-C mice, so just reduced allergic contact dermatitis in the mice that they got by treating one of their ears with a 1-chloro-2,4-nitrobenzene chemical, which is going to produce a reaction similar to that of, poison ivy or something other, nasty rash like that. So the RS extract was then administered during the sensitization and or elicitation phase. So they tested both of the phases to see, but yeah, they killed the mice, cut the ears off, and then they did a bunch of tests to them. So what they did was they used a special assay test. Like, I'm not going to read all of this because it's like a word that's 10 years long, but they looked at lymphocyte proliferation using a specific assay on the ears that they cut off of the sacrificed mice. And then they looked at the maturation of dendritic cells and differentiation of T cells using flow cytometry. Then they also looked at the mRNA expression of Tbet, GATA3, and FOXP3, which we know about, and evaluated it with PCR. Yes. So what they found was that the RS extract actually markedly reduced the swelling in the ears and also the cellular infiltration of inflammatory cells into the ear tissue. So then they looked at more, they did that assay that I talked about, and they looked and saw that the RS extract prevented the differentiation of naive T cells into Th1 cells, which reduced the inflammatory and immune response in the area. And the RS extract also reduced the proportion of dendritic cells in the sensitized mouse lymphocytes and in the expression of CD40, CD40 and CD86 cells in the dendritic cells.